What's up, everyone? My name is Kyle Lofi, and today we're going to walk you through a somewhat complicated process of meiosis. So, the purpose of meiosis is to start with a diploid cell and turn the diploid cell into 4 haploid cells, which will eventually become sperm and egg cells, also known as gametes. So each of these sperm and eggs are all going to be genetically different so that you can create a new organism when the sperm fertilizes with the egg. So this is due to a process in meiosis called crossing over and independent assortment. So in crossing over, as you'll see, these beads will represent DNA that is switched from the father part of the chromosome to the mother. So they will switch and it'll look like this. So this little part from the mom switched over to the part of the dad. And then to talk about independent assortment, we will talk about more later in metaphase 1 and metaphase 2, and it's when the chromosomes will line up in the center of the nucleus, the center of the cell, and they can be assorted in any way possible. They can be assorted right like this. They could switch over and back in any way they want. And when you have 26 chromosomes, that brings millions of different possibilities that could come from it. So, once these sperm cells and egg cells are made in your own body, the sperm and eggs merge after sex through a process called fertilization, as I was talking about, and the new baby will begin to grow, which will be genetically different from the parent through crossing over and independent assortment during meiosis, and that new organism with genetically different DNA is known as a genome. So, to start off with a little bit more background information, the white, the green, and the red parts of these chromosomes represent the mom, right? So they're going to represent the part of the chromosome that came from mom, and then the black, yellow, and blue parts of the chromosome will be represented from the dad. So, also, as I was talking about, these beads that will be put on later due to crossing over will be seen as what comes from crossing over, so the DNA being switched, which we'll go into more a little bit later. And then also Play-Doh, bits of Play-Doh you'll see in the middle, and that represents the centromere. And these wooden sticks will be seen as the spindles and kind of the centrosomes. So yeah.
Interphase 1 is very similar to interphase in mitosis, where the cell undergoes regular cell processes, grows, and replicates DNA. During interphase, the diploid cell has pairs of chromosomes, as seen here. It also grows centrosomes, which are later used during meiosis to pull apart the chromosomes. The S phase of interphase is when DNA is replicated, and now they are in pairs of the mother and the father. For instance, here, the black and white pairs are together, the yellow and green pairs are together, and the blue and red pairs are together.
Guys, this is the process of prophase, which is the part where the actual process of meiosis starts. So the nucleus is now broken down, so there's no more nucleus, and now the chromosomes are in empty space. And then so this is where the process of crossing over starts. So as you can see here, these beads are representing the new DNA of each parent being crossed over and interlinked with each other. And this gives the new organism a new variation. And each chromatid now looks completely different from their genomes, and it looks completely different from their parents, but they still have similar DNA from their parents. So yeah.
After prophase 1, we have metaphase 1, which is the chromatids, and they align in the middle, and they can be aligned or made anywhere they want, like that. And that is the law of independent assortment.
Alright, so now we're into anaphase 1. This is still a diploid cell, and so in this process, the chromosomes are being pulled by the centromeres and the spindles to the opposite sides of the cell, and it pulls the DNA to the opposite sides.
Alright, so now we're in telophase, and at this point, the cell is still a diploid cell until the end of this phase, where they will go to a haploid cell. So, in this phase, as shown here, each side has one part of the chromosome. So, it'll have one part of chromosome 1 over here and one part of chromosome 1 over here from the father and mother that have been genetically modified by crossing over. And so, the new nucleus has formed in telophase, but then in cytokinesis, this cell membrane represents the cytoplasm being splitted in these 2 new cells.
Okay, so post-cytokinesis, In interphase 2, we see there are now 2 haploid cells. In this interphase, they do not replicate DNA, yet they grow new organelles.
Okay, so now we are entering, after the interphase 2, we are entering into prophase 2. This is the 2 haploid cells. They were divided in cytokinesis, and then so there's no crossing over in this, and this is where the nucleus is disbanded in both cells, and the spindles are starting to form again, as you can see here, and the chromosomes are condensing, and the centromeres are still present in the cell.
Go.
Alright, so still haploid cells, metaphase 2, they start to align in the middle of the cell, and the spindles grab onto the centrosomes of the chromosomes, and they are aligned in the middle of the cell. Once again, the chromosomes could be flipped in any way, and are anyway providing many different types of assortment, but for the purpose of this video, we align them in a simplistic way, and yeah.
Perfect. 1.
Alright, so during anaphase 2, the spindles attach to the centromeres and pull apart individual chromatids to each side. They're still haploids, however now the chromosomes are separated into chromatids on each side.
So at this point, we're at telophase 2, and the chromatids got to their separate poles of the cell, and a new nucleus was formed around them. And then in cytokinesis, the cell membranes and the cytoplasms were split, forming 2 new cells from each of the other 2 cells we have, which means we have 4 cells now, starting from our 1.
No.
So at this point, we have 4 new gamete cells, whether that's sperm or egg cells, which we started from 1. And the importance of this step of meiosis as a whole is because, as you can see here, if you look at each of these new cells, they are all different. So they're all going to have different genetic code. So when they line up and they fertilize with the egg or the sperm, they're all going to be different, and that's going to produce a completely different genome. So when after the S phase and interphase, which we started with, we had 46 chromosomes and 92 chromatids. And at this point, in each cell, we'll have 23 chromosomes, but only 23 chromatids. Thank you for watching, and I hope you enjoyed our overview of meiosis.